Hi, everybody. We're going to take a minute to let Zoom allow everyone to flow into the webinar. Sometimes it takes a little bit to allow everyone who registered to join. I think we have probably most of the people here, but we're so glad you're all joining us today. This is a more important topic than I think people realize. So I'm grateful that you guys have joined us. Looks like we are about ready to start. I am Tara Haley. Welcome to today's webinar, Behind the Ebola Outbreak, an expert backgrounder for journalists, although we often have many others join us. I am the health beat leader on infectious disease for the Association of Healthcare Journalists, and AHCJ is excited to partner once again with the expert vaccine analysis team, a team of internationally recognized and trusted vaccine experts whose scientists and physicians have a long track record of leadership in vaccines, infectious disease, and public health. AHCJ is the largest organization of health journalists in the US, and we've partnered with EVAT to provide journalists on a regular basis with essential briefings about different topics. Joining us today, next slide, please, are Craig Spencer, an emergency room physician and an associate professor of the Practice of Health Services Policy and Practice at Brown University School of Public Health. Dr. Spencer contracted Ebola while working with Doctors Without Borders in Guinea during the 2014 outbreak. Daniel Jernigan, a public health leader and epidemiologist who previously served as the director of the National Center for Emerging and Zoonotic Infectious Diseases and director of the Influenza Division at the CDC. Mary Bassett, a professor of the practice of health and human rights in the Harvard T.H. Chan School of Public Health and previously the New York City Health Commissioner from 2014 to 2018 when she oversaw its Ebola response. And Philip Krause, who recently served as Deputy Director of the FDA's Office of Vaccines Research and Review, and previously served as a key advisor to the World Health Organization, including as chair of the WHO's Research and Development Blueprint COVID-19 Vaccine Expert Group. A little bit of housekeeping and a reminder that all attendees are muted during this webinar. While speakers are presenting, you are welcome to ask questions at any time by typing them into the Q&A space where the question mark is at the bottom of the Zoom frame. I'll direct the questions to our guests. I often jump around, make sure we try and ask all of the guests different questions. A reminder that a recording of this webinar will be publicly available today or tomorrow. Anything that's said during this webinar is considered on the record and may be used in stories or on other public outlets, excuse me. Dr. Spencer will begin with his experience in the 2014 Ebola outbreak and a comparison to the current outbreak. Go ahead, Craig.
Wonderful. Thank you so much for the introduction, and thank you for having me, and also thank you to everyone who is here and joining us. I want to start by doing just a couple things, which is looking at what we've learned over the past decade, talk about what's worrying me with the current outbreak and where I see some significant improvement over the past 10 years with the end of the 2013-2016 West Africa Ebola epidemic. You know, as soon as I heard about this outbreak just over a month and a half ago, I immediately knew it was going to be catastrophic, as I'm sure many others on this call did. But I also knew how much it sounded like what we had learned in 2014. If you remember, some of the initial accounts back then were about this outbreak that had likely been delayed in its detection by many months. It was occurring at the confluence of three different countries at that point: Guinea, Liberia, and Sierra Leone. And although it was kind of slowish moving at first, it started moving a lot faster than it felt like we could control it. Fast forward now to May 15th of this year, and we were told about an outbreak that was occurring in Eastern DRC. The number of cases reported on day one was already much higher than most outbreaks are when they end. It was also at the confluence of three different countries. And so we saw all along the eastern border of Eastern Congo, a place that I've worked a bunch in my professional career. We saw cases close to the border with South Sudan, just next door, which has an unfortunately weak health infrastructure where Ebola would likely do no favors. And then the same day, we saw cases and reports of a death in Kampala, the most populous city in Uganda, hundreds of kilometers away. Although there's been a lot of advances over the last decade in terms of treatments and vaccines, which we'll hear about in this call, we're essentially fighting this outbreak similar to the way that we were just over a decade ago. We have no vaccines, we have no approved treatments for the Bundibugyo strain of Ebola that we're managing here. And unfortunately, as I mentioned, the outbreak here is moving remarkably fast, and I still suspect that the case numbers that we're getting are not truly reflective of the actual size of this outbreak. Let me just take a minute to talk about the things that kind of worry me before I get into the places where I'm hopeful, and then briefly talk about some issues around the US response that I think need highlighting. When it comes to what worries me, I think it's important to note that 10 years ago, we had detection of cases in March 2014 in Guinea. This was delayed because we had never seen Ebola really in this region other than once, isolated different strain decades prior. Delayed detection meant that you were able to have cases in a bunch of different places, including in the capitals, but it took 6 months in West Africa to accumulate the number of cases that we already have in 7 weeks in Eastern Congo. So around, I think the latest report is somewhere around officially around 1,300 cases. We see the death toll picking up. And that has all happened in just the span of a month and a half, what took around 6 months for us to get in West Africa. And I think there's some differences in testing and capacity that don't make that a direct one-to-one comparison, but it should give you an idea in terms of the scale of what we're dealing with in Eastern Congo, why it is potentially so problematic. I would say that the best case scenario in my mind is that this ultimately becomes the second largest Ebola outbreak ever, I'm sorry, the second most likely scenario, bypassing the 2018 to 2020 Ebola outbreak in Ituri, the same region from just a few years ago. But I think the worst case scenario, and one that is still quite reasonable and possible, is that this becomes the worst outbreak ever and exceeds even the outbreak from a decade ago that had 28,000 cases. Projection by some WHO researchers published last week in The Lancet suggests the most likely scenario is around 8,000 cases and 1,400 deaths by September, so just in the next few months. And we're seeing a pace of growth every day with 30 to 40 new cases every day. I suspect that that is very, possible and likely reasonable. And a CDC set of projections included a worst case projection from a few weeks ago that suggested potentially 20,000 cases around the same period. The one thing that I unfortunately saw all too clearly while working as an Ebola provider in Guinea, and then experienced myself as a healthcare provider that was infected with Ebola as well, was that obviously Ebola does not infect indiscriminately. It's a disease of compassion, and it finds the people who care for others. In one MSF treatment center right now, 65% of the patients are women, older women that generally tend to take care of family members once they're sick. And healthcare workers are disproportionately impacted, 20 to 30 times more likely to be infected than the general public. A decade ago, this was important because there were as many healthcare providers in the one hospital where I was taken care of at Bellevue in New York City as there were doctors in Guinea, Liberia, and Sierra Leone combined at that time. And around 6 to 8% of those healthcare providers in those countries died during the span of that outbreak. This is concerning now in Eastern Congo because we're seeing the same thing play out. Congo has one of the lowest densities of healthcare workers relative to its population, with about 10 to 11 physicians per 10,000 people, according to WHO data. And we've already seen, according to Dr. Tedros last week in a WHO call, about 80 healthcare workers that have already been infected, according to the WHO just last week. The other place where I'm a little bit worried is that in the last decade, we've done incredible work of building out vaccines and treatments. And I know Phil and Dan will probably talk more about this. One thing that I often note is not brought up is that when I was treated for Ebola, I was lucky enough to be in a high quality center where I had intensive care with remarkably dedicated folks. I probably took care of 30 to 40 patients on a daily basis while taking care of Ebola patients myself. And there were probably 30 to 40 people on call every day to take care of me when I was treated. The inequities extend beyond that, however, in that over the past decade, we have created vaccines and treatments for Ebola Zaire, the different species of Ebola than what is behind this outbreak. Thankfully, we have been smart enough to create a stockpile of those vaccines where 500,000 are ready to go at a moment's notice to respond to outbreaks. But this is not the same for the treatments themselves. Two of the treatments, Ibanga and Inmazeb, have been shown to lower mortality. The problem is that they still largely remain inaccessible to patients, Ebola patients during outbreaks. That's despite the fact that one of these treatments was actually created from the blood of a Congolese survivor and received FDA approval after trials that were hosted in Congo. But during another Ebola outbreak just about a year ago, it was hard to get those treatments to Ebola patients in Congo because most of those doses are held and owned by the U.S. and require deployment. We are seeing a similar thing here in that there are investigational treatments that I think hold a lot of promise, MBP 134, particularly. We know that they are being deployed to be part of a trial. There are still questions and have been questions, justifiably questions, particularly from the Congolese side about post-trial access and any guarantees that these treatments will be made available and will be equitably available after the outbreak is over. Understanding the historical tendency has not been towards that direction. If one of these treatments, the MVP 135 by Matt Bio, works, it would be great because it's not just for Bundibugyo, it's not just for Ebola Zaire. It seems to be a pan-Ebola treatment, which would be incredibly helpful. This is important not just for treatment itself, but it's very hard to kick off an outbreak response and get people to come to Ebola treatment centers if you don't have a whole lot that you can offer. If you don't have testing that you can do with a quick turnaround. We saw very early on in this outbreak, people were going to treatment centers and then walking out days later because they did not have testing results. Low confidence, low trust in those centers, which makes it very hard to build buy-in. And just very briefly, before I pass the mic, I want to say the things I'm hopeful on here. The speed of response and the role that Africa CDC and WHO have been playing together along with the Congolese government have been truly remarkable. The cuts of the past year have been hard, but I am very, very heartened to see WHO and Africa CDC step up in this response. Similarly, the scaling of testing that has stepped up from essentially zero a month and a half ago to thousands of tests a day is incredible. It's not enough. And if this continues to grow at a similar pace, we'll need more. But this has been a remarkable improvement. And I am heartened to see that even if the US is not sending our incredible experts into Bunia, into Ituri, into the zone itself, we are contributing significant financial resources, 700 million, with another 1.4 billion apparently being asked for Congress to grow this response. I think there's a lot more that we can be doing on the ground, and where the absence of the US over the past year and really even over the past month has been problematic. But I will leave with that and I will pass it back to you, Tara.
Forgot to unmute myself there. So thank you very much, Craig. I'm now going to turn it over to Dr. Jernigan, who will discuss his experience coordinating the in-country responses and the U.S. national response in 2014.
Great. Can you hear me and see my slides?
Yes.
Okay, great. Thanks very much. And so, so I was the director for the National Center for Emerging and Zoonotic Infectious Diseases, but also in 2014-15 served in Sierra Leone while Craig was over in Guinea, and then came back and served as the incident commander for the Ebola response at headquarters in Atlanta. So if we go to the next slide here, if I can make that happen there, just a little bit about some of the background. And I think Craig got into a little bit of this, but there are these three countries there. Sierra Leone, largely an English-speaking country, where you've got recent civil war and a lot of unrest and uncertainties there with trusting the government. Liberia, highly correlated with the United States. That has also had a series of problems with civil war and others. And then Guinea, the French-speaking country that just sort of comes up and over both of those in West Africa. If you see where the star is, that's Gekadu. It's where one of the first patients was identified, and it was a journalist that actually went in and really tried to figure out exactly who that was. And thanks to that person for seeing that. But some bushmeat probably eaten, some infections occurring, other people getting sick, and then people moving out from that area to seek care. Initially seeking care with a traditional faith healer or a traditional healer in Kenema, which you can see one of the little cities down there in Sierra Leone. A huge number of people went there. The traditional healer died. A lot of people went to that person's funeral. A lot of people got sick. The hospital was overwhelmed there. And so that really kicked off a whole lot of cases that then moved into those urban settings. And so I think what's important is some of the stuff off there to the right, where this whole area has weak public health infrastructure. There was limited clinical and lab capacity with no real experience with Ebola. They had very porous borders, and the population was very mobile. If you went in to try and do some intervention in one area in northern Sierra Leone, they would just move over into Guinea, where you would have not the same connections and with dealing with a different government. So it was very difficult. The dense urban transmission was really the unique thing here, where people were sharing their Ebola very easily within that urban setting. It became very difficult to control. The distrust of government and rumors, and again, like there were no vaccines, drugs, or tests. And the final being 28,000, 11,000 deaths. And the epidemic curve down to the left there just shows you what happened. And you can think about that amount of time that Craig was just talking about, where we might be now. Look at the size of those curves that follow that period. So the worst is yet to come if the same process is going to be following with this particular outbreak. So there are some things that happened, things that were good, things not so great. But the whole response was set up as pillars. And this really did not take off until you had overall international collaboration and coordination through UNMIR, which was the United Nations Mission for Ebola Emergency Response, which coordinated the other WHO and other UN agencies like WHO and UNFP and World Food Program, etc. The four main components were case finding and contact tracing. And as you heard from Kaseya from Africa CDC, he's very concerned that we're not doing contact tracing right now at all, enough to really keep up with the numbers of cases. That little hut there with the blue roof on it is where we would take people in Sierra Leone. They would possibly get testing if it was available. They'd sit there until the result came back inside the little fenced-in area. Off to the right is the quarantine monitoring that CDC did in West Africa. You can see it's pretty porous. People could just step away from that quarantine if needed. In terms of case management, there's a photo there from one of the Ebola treatment units, a Uganda physician in that picture. From laboratory, we actually use the United Nations Humanitarian Air Service to move specimens around. So what you're taking home here is an enormous amount of coordination, international collaboration, et cetera, that has to be in place. Safe and dignified burial, absolutely critical. This was a big problem then. It is a big problem right now. You have to work with anthropologists, with social media people now, with key opinion leaders, imams, etc., in order to make that work. But one of the more important things is this social mobilization and community engagement. You can see a statement there in Krause from Sierra Leone, but you have to understand what they want, what they need, and understand what it would take for them to adhere to something that they are very suspicious of in terms of trying to control Ebola. In terms of the United States domestic response, we just didn't have that many cases, but it really started off in September of 2014 when a man came back from Liberia into Dallas, went into his emergency room, had some non-specific symptoms, but said, I was in Liberia at that emergency department. And he was given some antibiotics, told he had sinusitis, and went home. Several days later, he was brought back very sick, was admitted, and eventually there were two nurses that did get Ebola from that context there at the healthcare facility in Dallas. So that one really demonstrated that what we were saying at the time, which was anybody can take care of Ebola, and even some leaders saying it's a wimpy virus, that those kinds of statements became people were very suspicious of them after this event, and it created quite a lot of panic in the media, etc. And so it did lead to some changes. Overall, there were 11 Ebola patients that were cared for in the United States. You've just met one of them. And 9 of them got their infections outside the United States, and 2 got them inside. The cost for one of those cases, I won't say which one, has been estimated at $4.3 million, which is exceedingly high. The response to the Dallas outbreak may be $20 to $25 million overall because of lost hospital revenue. It revealed here vulnerabilities in recognizing things at the clinical front door. So we had to change what we were communicating to physicians about what they need to be looking for and how they need to be thinking about Ebola. Moving to safe specialized care. So Emory and Bellevue and Nebraska then started taking on specialized Ebola care instead of other people trying to do it. Doing credible and risk communication so for the public wasn't just hearing that you're not at risk, but they were hearing more about what the potential risk is and about what it means to go into a healthcare setting in the era of Ebola. Travel restrictions were put in place. We funneled flights from all the affected countries into 4 airports in the United States. About 29,000 people were monitored by health departments in the U.S. A significant amount of countermeasure development ramped up with new vaccines, new drugs, new therapeutics, point of care diagnostics. And so all of that really allowed those things to be used to control the outbreak. And frankly, the use of ring vaccination really probably was the main lead for getting it under control. We stood up the new National Ebola Treatment and Evaluation Centers and a huge amount of funding, about $5.4 billion the federal government requested, and about 1.1 billion of that was for domestic, and a fair amount of that was actually to pay for people taking care of screening at the borders. The pictures at the top are from Duncan's house, the guy that was in Dallas, and the stigma that sort of generated from that event was the second one is folks being taken into Emory, and then the third was a general sense from the public that you should ban all people coming into the United States. And so those are issues we're dealing with now. Just to finish, in terms of current challenges, the United States government funding, I think, is in the right direction. Craig, you mentioned that. They're looking at big numbers. That's good. But the USG leadership is not. If you look at what Tom Frieden was doing back in 2014-15, he was absolutely vocal and really made WHO look bad because they needed to get out in front of things. And I think he really helped bring folks together and got a lot of focus on that. And then, of course, with international engagement and with the UN getting involved for coordination, things got better. The current 25% staffing cuts at CDC and the increased bureaucracy to move all transactions through politicals will stall and slow needed deployments and response efforts. The loss of USAID presence in Africa, I think, hampers the response. It probably may have prevented earlier detection of it. The separation of WHO makes for very difficult collaboration right now. And then not repatriating Ebola infected or Ebola suspected Americans is bad for response volunteer participation, and it disregards use of the U.S. government's U.S. government's funded world-class treatment centers at Emory, et cetera. And so to finish, I had talk to somebody at one of those centers, and they said, we're not getting these folks because they say it's safer for them to be taken to Kenya or to wherever. But in fact, they're using the same medical transport, the same one, Craig, you may have been on with Phoenix Air, to send our countermeasures over there to be used rather than sending those folks that could be cared for here in that same transport. So with that, let me hand it back over to you, Tara, and I really appreciate the opportunity to talk.
Thank you for that, Dan. Very sobering. Next, we'll hear from Dr. Bassett on what it's like to be on the ground responding to it and communicating during an outbreak in the US.
Thanks very much, Taryn. It's a real pleasure to be here. I've really benefited from my connections to EVAT as as the New York State Health Commissioner, and I'm very grateful for this connection today. Actually, the last time I saw Dr. Spencer was probably just a few months after he had recovered from Ebola. So I'm going to talk about having been the New York City Health Commissioner at the time that we were beginning to plan for the likelihood that somebody with Ebola virus disease would appear in New York City. As you've heard, all of the returning people who were visiting, who had visited the three affected countries, were funneled through certain airports, and New York's area airports were one of them. And what I think might be important that might not be reduced to dollars and technical supplies. You've heard that this response, New York City's response, was expensive. That's true. The New York City Health Department spent $4.3 million in responding to one person and undertaking the other responsibilities that we had. 114 health workers had to be followed and monitored, and so did all the people returning from having traveled to the affected countries. But important to me were a couple of things. One is the fact that preparedness is not simply a matter of having enough stuff and enough money. It's also a matter of having the right relationships and having actually practiced the procedures. So there were people who seemed to think that tacking a sign up about how to don and doff personal protective equipment up on a wall was enough. And fortunately, for everybody in New York, the experts at the New York City Health Department did not think that. So the city began planning for the possibility of a case even before WHO had declared the Ebola emergency in July of 2014. We had some assets because of the AIDS epidemic, which New York City was an epicenter for. We had a special treatment facility for people with multi-drug resistant TB with its own laboratory that was reconfigured to be a place where we could look after somebody with Ebola. All of the hospitals in New York City were supposed to be getting ready, but I don't think it's telling anything outside of school to say that the only hospital that was ready at the time Dr. Spencer was diagnosed was Bellevue Hospital, which many of us consider sort of the crown of the very robust public hospital system that we have that we had in New York City. So we had to work across agencies. Not only did there have to be coordination and figuring out how to make sure these worked effectively between the health department, which was going to do the diagnostic tests through its underfunded to this day public health lab, had the capacity to test for Ebola. We had to coordinate with the public hospital system. We had to coordinate with the fire department that ran the ambulance service. And we had to coordinate with outside entities, the UN agencies, MSF. We had a protocol for the identification and reporting of any person who might need to be under investigation for Ebola virus disease. So this kind of coordination is not just something that works when you have a rule book that says how it should be done. You have to practice it. You have to refine it. And fortunately, at the end of October of 2014, the city was in a really good position. The sanitation department had to be involved in waste management. There was a lot of coordination within government and with entities outside of government. Um, and in addition to that, there was the whole question of public communication and talking with people about the fact that we had somebody who'd been diagnosed with Ebola, who'd ridden on the subway. Um, and I have to say, after our first press conference, when I took the subway to work the next day, I thought to myself, I don't even know if there'll be people on the subway. But fortunately, people believed us when we said that the subways were safe. And probably one of the things I'm most proud of is our special outreach to the communities that had ties to West Africa. There's a large Liberian community in New York City. We had town meetings with them. We not only wanted them to tell us if they were being mistreated simply for their national identity, we also wanted to give them information about Ebola virus disease because they might be in better contact with people who needed this information than their own governments. And we also had to do things that we didn't expect. For example, we had maps printed up of where these countries were. Some of them you saw the map that Dr. Jernigan just showed. We put those up in emergency rooms all over New York City. We pointed out that Guinea and Guyana were not the same place because that had happened. The mantra was travel with fever, and unfortunately, somebody got hauled off because they were from Guyana and had a fever. Um, and I also think another thing that will be important as we talk about the risks to people in the United States, which were then and are now not high, that we continue to reiterate that there has to be control of the virus at the source. That none of us, if there's ever a time that we learn about our global world, it's with highly contagious viruses like this one. And we should make it clear that not only do these countries need stuff, the Eastern Congo needs peace. The only way that they will be able to manage this truly effectively is if somebody can build up the trusting relationships so that the warring factions that have been feuding for many, many years in that part of the Congo sit down and understand that they have a joint obligation to protect their populations. The direct costs to the health department were about 4.3 million, but the cost of the city was about 20 million. So these are costly undertakings. But I think for me, the biggest take home really is the importance of processes and people in addition to technology and funding. So I am not involved in the current response, but it sounds, and you've heard from the two previous speakers, that the decimation of our global health investment, the separation from the World Health Organization, the severe cuts to the Centers for Disease Control, which have trickled down to severe cuts to state and local health departments, can't be helpful in ensuring that we're prepared. And with that, let me hand the mic back.
Thank you, Mary. Finally, we're going to hear from Dr. Krause on therapeutics and vaccines for Ebola Bundibugyo. And I suspect Dr. Krause can talk about whether or not the current vaccine could be adapted for the current species.
Sure. Thanks, Tara. So let's start off with a little bit of the taxonomy of these viruses, because how they're related to one another is actually quite important in thinking about what therapeutics and vaccines might work under the current circumstances. And Bundibugyo and Ebola virus Zaire are different viruses that are related but represent different species within the family Filoviridae. And then they're also in the same genus, Ebola virus. So there are three main viruses, Sudan, Zaire, and Bundibugyo, that are in this Ebola virus genus, and then there's another one, Marburg, that is in a different genus. So Bundibugyo and Zaire is the virus that caused most of the big recent outbreaks, including the ones that have been discussed today, are related because they're in the same family and genus, but they're actually different species of virus, which means that they're in many ways pretty different from one another. It's not like they're different strains of the same species where you might expect there to be a lot of crossover protection. And in fact, the disease with Bundibugyo is a little different from that with Zaire as well. And while the symptoms tend to be very similar, on average, the Bundibugyo disease is not as severe as Zaire. And so you see, on average, lower case fatality rates, longer time to death with Bundibugyo than with Zaire, but there's an overlap. And so the mildest cases of Zaire might not be as severe as the most severe cases of Bundibugyo. But of course, so far, according to the WHO situation report, on June 17th, we have 896 confirmed cases and 232 confirmed deaths, which gives you a case fatality rate in the 30s. And probably when all the data shake out, it'll be a little lower than that, but this is still a very bad virus to be infected with. And of course, this particular outbreak, as Dr. Spencer has pointed out, is made that much more difficult to deal with based on where it is and the warring factions and the difficulty of access to these sites, especially in the Democratic Republic of the Congo. So what do we know about treating these Ebola virus infections of different types? Well, the first thing we know is that one of the most important things you can do is provide supportive care. And so having modern intensive care units where you can manage people's fluids and make sure that they don't die of things that otherwise could be treated, electrolyte disorders, things like that, is one of the most important things for keeping people alive. And so that's a critical component of treatment no matter what you do. For the Zaire virus, because of the previous outbreaks, so it's the opportunity to test a number of different drugs. And they really fall in two categories. One of them is monoclonal antibodies, which are designed to bind to the surface glycoprotein of the Ebola virus and either neutralize it or cause a different immune response that will stop the virus in its tracks and will treat the infection. And they're actually in the previous Congo outbreaks, 3 monoclonal antibodies were tested. One of them is Inmazeb, which is a combination of 3 monoclonal antibodies, and another is Ibanga, which is a single monoclonal antibody. And both of those had pretty good efficacy and clearly reduced mortality. And then there was ZMapp, which was another cocktail of 3 monoclonal antibodies, which it turned out didn't work as well as those other 2. And so it isn't the case that just because you come up with a monoclonal antibody that binds the virus and it looks like it works in animals, that you're sure that it's going to work well in humans. So the clinical trials really needed to be done to determine that those 2 monoclonal antibodies, the Inmazeb and the Ibanga, actually worked. In these same studies, another drug called remdesivir was studied as well. And remdesivir is a drug that inhibits the RNA polymerase of the virus, which the virus needs in order to make copies of itself and to replicate in humans. And you may remember that drug because it was also used for COVID. So the advantage of a drug like remdesivir is it can be very broad spectrum and can treat different viruses as long as the RNA polymerases are similar enough. And so this is one where there's the potential that it has broad spectrum, and whatever activity it has against Zaire, it might also have against Bundibugyo. But the problem is that in the clinical trials earlier on, remdesivir did not work as well as the monoclonal antibodies did. So I guess what I want to say here is that it's very important to do these studies to figure out what works and it doesn't work, and make sure that you do this in a setting where you have randomized people and understand what is happening, because unless people are getting exactly the same, have exactly the same likelihood of getting similar supportive care, you might end up being fooled regarding what's working and what isn't working. Now, there's another very intriguing monoclonal antibody called MBP-134. This monoclonal antibody was designed to cross-react with shared components on all of these Ebola virus genus glycoproteins. And so because it can bind to Bundibugyo as well as to Zaire and even against Sudan, there's the hope that this might turn out to be a treatment that could cover all three of these viruses, while the other monoclonal antibodies were designed only for Zaire and would have no hope of being able to treat Bundibugyo or Sudan. And MBP-134 was given to people on a recent Sudan outbreak, but we don't have any information on efficacy. And like the other monoclonals, it works pretty well in animals, but we really need studies to figure out whether it's going to work in humans. But there is some hope there. Now, what about vaccines or the settings of post-exposure prophylaxis? Here we've had enormous success with Zaire because the vaccine Ervebo, which is licensed in the US, is a single-dose vaccine, and it's a different virus called vesicular stomatitis virus, which is made to express the glycoprotein of Ebola virus Zaire, induces pretty strong immune responses against Zaire. And it was shown that if you can give that vaccine to somebody, even after they have been exposed to Ebola Zaire, you can actually prevent, with fairly high efficacy, them getting Ebola virus. So that gives rise to an amazing ring vaccination strategy where people get exposed because they had an exposure to a case. You immunize them soon afterwards, and you actually can protect them. Now, those who were already infected at the time you immunize them, you can't protect those people. But starting about 10 days after immunization, you don't see any cases. And so that can play a huge role in stopping an outbreak. The problem, though, is that Bundibugyo, as well as and Ebola Zaire, have different glycoproteins. And while there's enough similarity that if you carefully design an antibody like MBP-134, you can find some cross-reactivity. Most of the immune response to Ervebo is against the Zaire virus, not against the Bundibugyo virus. And so the expectation that Ervebo would work against Bundibugyo is actually pretty low. Now, this has actually been studied in animals, and 2 experiments have been done, both of them with not enough animals to draw any conclusions. And in one of them, there was a very modest suggestion that it might help, and the other one, it seemed clear that it didn't help at all. And so we don't really have much help from the animal experiments suggesting that it would work. And we don't have a lot of hope based on what the vaccine is and the kinds of immune responses it induces and the similarities and differences of these viruses that it would work. And yet this can be studied. You could take people, for instance, who in the past had received Ervebo and ask whether previous receipt of Ervebo might influence their likelihood of getting Bundibugyo, and that can be done even without immunizing any more people, because a large number of people have been immunized with Ervebo. There's another vaccine against Ebola Zaire, which was made by Janssen. It was a 2-dose vaccine that was where the first dose was an adenovirus vectored vaccine, and the second dose was a vaccinia vectored vaccine. So both other viruses that were engineered to be both very weakened, but then also to express Ebola glycoproteins. That, in order to get strong immune responses, that vaccine required 2 doses. And in general, was never tested in humans, although it worked well in animals and humans got very strong immune responses to it. It had some hope of being useful. And that was actually licensed in Europe and was pre-qualified by the WHO, but it wasn't licensed in the US. And that vaccine is becoming less and less available, although there are still some doses of that. But that's also a vaccine that doesn't have any significant hope of covering Bundibugyo. So where do we stand then with Bundibugyo? Well, there's, I guess, three possible candidates that have been put forward as leading candidates. There are other things that people are trying as well. One of them is to take the same VSV virus vector that is used to make Ervebo, but instead of having the Zaire glycoprotein in it, put the Bundibugyo glycoprotein in that. And so that's an approach that we know has worked for Bundibugyo. And so the hope is that that vaccine could work. But of course, we'll have to test that to see whether or not it actually does. And sometimes when you manufacture these vaccines, they don't work as well as you hope they will, just based on the pure science. There's also a chimp adenovirus vectored vaccine, which is based on the same backbone that was used in the AstraZeneca COVID vaccine, which expresses the same Bundibugyo surface glycoprotein. And so that's something that could potentially be tested. And then there's Moderna has a vaccine, which is an mRNA vaccine that expresses the Bundibugyo surface glycoprotein. So these are all vaccines that have some hope of working. But what I can tell you about vaccines is that all of these are in fairly early stages of development. We may have doses of some of these vaccines available in a month or so, but those would still need to undergo testing in phase 1, which would take probably a month or two. Before then, they could be tested to see whether or not they're working in the outbreak. And so we're hoping that the outbreak gets under control faster than that. So we don't know whether it will be possible to test these vaccines in the outbreak. But of course, everybody's looking towards that and wants to plan to do that in case the outbreak does not get controlled by then. There's one more promising approach that can be considered for preventing Bundibugyo disease in people who've been exposed, which we did not have available before. And that is that remdesivir, I told you, is an antiviral drug that has activity against all of these viruses, but it did not work as well in treating established Ebola disease, Ebola Zaire disease, as the monoclonal antibodies. But one of the reasons for that is often because if you give antivirals after disease has been established, there's a lot more work for them to do because they have to, the main thing they can do is prevent further replication of virus, but they can't necessarily do that much about the virus that's already there. But in a person who's only been exposed and maybe doesn't have as severe an infection, if you were to give them an antiviral drug very, very early in the course of the disease, there's a chance that you could prevent the establishment of disease. And for remdesivir, there's actually an oral homologue that is made by the same company called obeldesivir. And so it is reasonable to suggest, at least, and to test whether if you would give somebody obeldesivir after an exposure, whether you might be able to prevent the establishment of disease in that person. And so all of these are things that people in Africa and the WHO are thinking about and are working towards conducting clinical trials on, so that the next time there's an outbreak, we're better prepared. The one thing that is disappointing is that we know that these outbreaks occur. And it's a shame that we don't yet have doses of vaccine ready to go in these studies right now, and the companies are scrambling to produce these doses, which means that the testing will be delayed relative to the trajectory of the outbreak. But nonetheless, there's a concerted worldwide effort to try to address these problems now, and I think it's only going to be with more attention and greater funding, and hopefully learning some of the lessons of the past, that will be even better ready the next time an outbreak like this occurs. But thank you.
Thank you, Phil. Excuse me. Thank you, all of you, for giving us all this great information. It's quite the all-star panel we have here. I'm grateful for that. We only have about 10 minutes for questions, and we have a couple there. So I'm going to ask the panelists, when I ask the questions, try and keep your answers real brief so we can get through them. A question from Dan. And Dan, I hope your question was answered regarding the species and strain. If not, let us know. But in terms of supportive care, is the major effect of Ebola its hemorrhagic effect, or can that be addressed passed, like abrogated? And I guess that's probably a question for Craig.
Um, so this is a little bit tough because, well, let me try to be succinct here. In certain cases, yes, the hemorrhagic effect is the primary concern. So I'm thinking in women during delivery, or in patients who have profound hemorrhagic symptoms, which may include GI bleeding. But what is actually more problematic is usually metabolic or electrolyte disturbances, so renal failure, kidney failure, sepsis, also superinfection. And so we saw that there was an increase in mortality, for example, in people who had Ebola and malaria, which is why we treated cases of malaria, pneumonia, etc. So it sounds like, according to recent reports, the number of hemorrhagic symptoms for Bundibugyo is less than it is for Ebola Zaire. I'm a little circumspect as to how true that is, quite frankly, as someone who collected the symptoms in 2014, and I can tell you people weren't particularly comprehensive about looking into everybody's eyes or looking at their gingiva if they're bleeding. I took care of hundreds of Ebola patients. I did not see all that many that had frank, obvious, extensive bleeding. Red eye, sure, gingival bleeding, hiccups was a really big concern. I think what we're seeing the majority of the time is that there's electrolyte abnormalities, dehydration, plus kidney renal failure that impact cardiac function and and respiratory function as a primary cause of death.
Thank you. Is anyone at the NIH still working on any of these vaccine candidates? That's probably a question for Phil, I'm guessing.
I don't know enough about what's going on at the NIH. The candidates that I'm talking about, though, are much farther advanced than the research stage. These are all candidates that obviously companies have picked up and have started manufacturing doses for testing. I'm sure that there are people at the NIH who are looking at other candidates that are not as far along, but I'm not up enough on that to be able to say what the current research portfolio is.
And a quick follow-up, what's the difference between a species and a strain in this context?
This is something that virologists try to figure out based on the total amount of sequence similarity, the genomic structure, and everything else. So the structure of the genomes, the proteins and genes that the viruses make is pretty similar, even among viruses of different species. But very often the disease that viruses of different species cause can be quite different. I can give you another example. Herpes simplex 1 and 2, for instance, are different species. They aren't different strains. And they actually cause different diseases, right? Herpes simplex 1 is more likely to cause oral or ocular herpes, and herpes simplex 2 is more likely to cause genital herpes, although there can be some overlap too. And that's a separate webinar that we can do another day.
Is it like a certain threshold of genetic difference that makes the difference?
It's mostly genetic difference. That's correct. But not just the RNA and DNA of the virus, but it's what do the proteins look like? How similar are the proteins? And often the proteins will even have somewhat different structures between different species within a family, not just different sequences in spots. So there's substantial differences in the protein and the structure, and therefore the antigenicity and the immune response to all of these viruses if the species are different. Whereas for strains, they're much more likely to be overlapping.
And I'm going to quickly, I don't know what our audience level's knowledge is. Antigenicity basically refers to the proteins being an antigen, which is the thing that the body recognizes for the immune system to mount a response. So if your proteins look different, then what antibodies you need to recognize those proteins looks different, which affects what kinds of vaccines you can use. I'm just throwing that out there for anyone who might not be familiar with that. Dan, you mentioned if the same process follows is like, look at what those huge waves look like. Is it still possible right now to prevent that? And if so, what would it look like? Or is it really not possible? We're going to do what we can, but that's still what's probably coming.
Yeah, I think it's going to be very difficult. The main thing is that the U.S. federal government can't stop that kind of a curve by closing the borders. They have to get in there and really make it happen. And so right now, the estimates, even coming from CDC, would put it at somewhere between 8 to 20,000 of cases. And so if you can't get into the urban settings, if this gets into the urban settings and you don't have good control of that house to house stopping and checking to make sure that people don't have Ebola, you're not going to be able to control it. So I am deeply concerned that with the current level of effort, we will see a rise, just like we did in 2014-15.
Um, Sort of a combined question, and I'm not sure who the right person is for this, maybe Dan. Why is the US hoarding treatments right now? And what is the US actually doing? Or I guess what should we be doing? What could we be doing that we're not doing? I know it's a loaded question and you could probably talk all day on it. So
Yeah, I'll others can answer that too. Let me just make one point about the availability of new vaccines. I do want to point out that $500 million of mRNA vaccine technology was cut. And so that's 500 million that could have been put toward a new mRNA for one of these emerging problems as well. The issue with the U.S. government's development of drugs through the Biological Advanced Research and Development Administration, BARDA, that's a group that actually helps develop these things that don't have a market, and so they stockpile them and they can hold on to them, and those can be released, for different things like we did for Mpox and for other things. So there's a role for keeping things as a stockpile for these rare events, and I know that some of the materials are being sent over to Africa. And I think one of the monoclonals is actually being used for a trial there. So the federal government is helping out in that way. But it is sometimes very difficult to get some of these experimental therapies to other countries, just simply because the regulations in those countries sometimes prevent you from actually just dropping them in and getting them treated. Craig?
Yeah, I think that's fantastic. Thank you, Dan, and thanks for your leadership on all this over such a long period of time. I want to mention two things that I think are pertinent here. One is there are apparently at least two cases of Marburg virus disease in Western Uganda that have been going on, it sounds like, for the last week or so. So Marburg, very similar to Ebola. This is horrible timing, but the reality is, I think with climate change, with increasing human contact, with whatever the reservoir is here, we are seeing a very significant increase in the number of viral hemorrhagic fever outbreaks. I don't think that's just an increased ability to detect them, although we have that as well. The fact that we have a Marburg outbreak layered on top of what could be potentially the worst Ebola outbreak, I don't think is coincidental. I think this is going forward going to be the reality. So I think we need to think about that in terms of our response, our medical countermeasures. And the one other thing I want to note is that my best friend was working in a hospital in Rwanda a year and a half, 2 years ago, and called me and said, I think we have a viral hemorrhagic fever outbreak here. This was 2 days before Rwanda declared a Marburg outbreak. I spoke to someone in the administration who, within 8 days, and based on, CEPI having just the week before done a roundtable and having good capacity and relationships in the White House with Rwanda, was in the span of 8 days able to get investigational vaccines and treatments from the US into arms in Rwanda. And that outbreak ended with the lowest recorded viral hemorrhagic fever mortality ever at around 22%. And Rwanda is certainly different than Eastern Congo, despite proximity. Their healthcare structures couldn't be much more different, but it did show the potential that we have as a country in terms of mobilizing resources, the speed and the strength that we're able to do that. And to Dan's point, money is not going to stop an outbreak. Building a wall taller and thicker is not going to keep it out. The only thing that is going to do so here is putting the outbreak out where it's at. The longer we let it linger and grow bigger with 10, 20, 30,000 cases, the increasing risk it will represent here at home to having sporadic or more cases.
A quick legal question. Do you guys know, similar to the situation with the viral outbreak on the cruise ship, can the US government legally prevent a citizen from coming back into the country?
That's a constitutional question that somebody else, I think,
don't know.
I've heard constitutional lawyers say that they can't. But anyway,
The question is, how do they get back here?
yeah.
And I think this has been the question with this facility in Kenya. So I think again, I'm not a lawyer, but what I've heard when speaking with lawyers is that no, they can't necessarily keep you out, but the question is, how do you get in? If you can't get on a commercial flight, who's going to get you here? And what does it look like in terms of the contracts with Phoenix, whether this is DoD, state, et cetera? So there's a big difference between what's right and what's actually done.
Yeah.
Don't really have time, but I'm gonna ask one more quick question. This is for Craig. What does that loss of healthcare personnel that you were referring to and caregivers in these countries mean in the long term? Like, can you kind of spell out the implications more broadly that people might not be thinking about what that really means, sort of countrywide?
Yeah, briefly, look, I've seen so many AI-enabled programs for biosurveillance that are going to detect the next outbreak. I spent years of my life living in Eastern Congo in Burundi. I did a bunch of surveys. I went to every single clinic. I talked with folks. I saw empty clinics. We are not going to be able to like engineer our way into empty clinics where red flags need to be raised. The CDC had a training program that ultimately trained a community health worker in Tanzania 2 years ago who recognized a Marburg outbreak and notified the government authorities, and that was put out quickly. We can use tech. The reality is that we just need to fund the bread and butter stuff on the ground, people in clinics that understand what Marburg and Ebola look like, and then have capacity to step that up. Much of that has fallen apart over the last year, unfortunately, primarily because of a pullback in US funding and support. And I worry that we're going to learn the lesson again we've already learned already and forgotten, is that we just need that infrastructure in place. And without it, not only are more outbreaks like this inevitable, but the likelihood that they impact us financially, but also potentially in a greater healthcare aspect is much greater. So we need those folks on the ground. There's no way around it.
Like I mean, when you lose those healthcare personnel, you also lose the people who are vaccinating kids and taking care of pregnant women and all of the other non-emergency things that happen in general. Thank you, all of you, for sticking with us. Thank you to the speakers for tuning in today. We really appreciate it. If you joined us late, we'll be sending everyone the webinar recording and post it to YouTube within a few days. And again, thank you so much for sharing your expertise. I'm really astounded by all the experience we have here with us on this. Thank you, everybody. Good luck with your stories if you're writing them. Take care.
Are we off?
We're supposed to be. We are.
No, I just thought that was a great idea about looking at the experience of people who've been immunized for Zaire and seeing what they're. Somebody should pick that up. That's a very good idea that Phil mentioned.
Yeah. There was, I was trying to keep up with everything. I had I didn't even get to ask.
Yeah. Anyway, we're probably being recorded. Thanks very much. But that was such a good idea. Somebody should pick it up. Yeah.
I'll edit this out of the recording. Thank you both
Thank
very
you.
much.
Okay,
And I'll close
bye-bye.
it down now. Have a great afternoon.
Thanks.
Bye.